Let's talk a little bit about stem cells, progenitor cells. These are specialized types of cells that exist, and we need to touch on what they do. So differentiation is the process of making specialized cells, going from a stem cell to a mature cell that can do what we want it to do. A liver cell, for example, would be a highly differentiated cell. Most of the cells we'll talk about all year are highly differentiated cells, but they all began as stem cells. A stem cell has the ability to form two new cells, one of which will be a stem cell. That's called self-renewal. One of the stem cells, resultant cells, can also become a specialized cell. It's a progenitor cell. That cell itself won't be a liver cell, but it will be something along that will continue to divide and move down the pathway to become something like a liver cell. We can differentiate into many different cell types. There are two basic kinds of stem cells traditionally: the embryonic stem cell that can become almost anything, or actually can become anything, and the adult stem cell, which is more a progenitor cell that can become part of a specialized tissue. So progenitor cells are partially specialized stem cells, are daughters of stem cells. They can become into a restricted number of cells so they become committed cells. Totipotent are daughter cells that can become any cell type. So a totipotent cell would be what we call an embryonic stem cell. So right after fertilization of the egg, and the early cells of the blastocysts are totipotent stem cells. And whereas adult stem cells are now called pluripotent stem cells, they can become a limited number of cell types. Stem cells that are evolved in later development are progenitor cells. They make specific tissues like blood tissue and liver tissue. So we got a stem cell progenitor in the formation of blood. So we've got a stem cell that divides, and that replaces the stem cell that's was there. The other cell becomes a progenitor cell. In this case, it's a myeloid progenitor cell, which is going to divide again and become one of two specialized types of white blood cells, depending on what happens as it divides or when it divides the second time. That process is called differentiation, making specialized cells. A fertilized egg has the ability to produce any type of human cell. It produced all types of human cells. So the fertilized egg divides to give us stem cells. Those stem cells divide and divide to give us an early blastocyst. They continue, then some become progenitor cells, and those progenitor cells become more differentiated until they reach their type of cell that they're going to become. Stem cells can be used to study. They can also be used to treat disease. There's a lot of work going on, some more successful than others. But in order to do that, and because of this, we have a field emerging called regenerative medicine, using the body's ability to generate new cells to treat disease. You can use donor stem cells, such as umbilical stem cells, or stem cells from the patient themselves, either from the natural site, such as the bone marrow. And I have a very good friend who, because of a cancer in his bone tissue, had to have a bone marrow transplant. They took his bone marrow out, got rid of the bad cells, and put the good cells back into his bone marrow after they wiped out his bone marrow. And he has some of those stem cells stored away now. And they can be reprogrammed in culture on occasion. There's some things that can happen to cells. One important one, especially during development, is called apoptosis. It is a programmed cell death. Again, it's a normal part of development. It's a stepwise process. It gets rid of tissues we don't want. For instance, the tissue between our toes and fingers was removed in the process of development by apoptosis. It's also a way to get rid of damaged skin cells after a sunburn. It's much less inflammatory than other types of cell death. Necrosis is cell death from damage. That is not a normal process, and that is going to lead to an inflammatory response, which, although is good in some cases, can be damaging in others. Proteins are synthesized in the rough endoplasmic reticulum, fats in the smooth endoplasmic reticulum. Smooth endoplasmic reticulum also is where sugars are built. Sugars and proteins are released from the Golgi vesicles and secreted from the cell. Fats go directly from the smooth ER to the cell membrane, and they're encased in cells, membranes, and exported. This is that endomembrane system that I went over with you on